Hello, everyone. I hope you are enjoying your life. In today's video, I will talk about the topic that is classification of microorganisms. I hope you will see this video completely and like this video because you will definitely learn a lot from this video. Let's start the video. Microorganisms are classified into acellular organisms and cellular organisms. Acellular organisms. These are the microorganisms that have neither cells nor cell membrane. They are composed of few genes which are protected by protein coat. Outside of the host they act as dead. But inside host they are alive and reproduce. Example: viruses. Cellular organisms. These are the microorganisms which have definite cell structures. Based on the cellular structure they are further divided into prokaryotic and eukaryotic. Prokaryotics are cells without nucleus and have no membrane bond organelles. They are less complex in structures. Examples: bacteria and archaea. Next is ultrastructure of bacteria. Under electron microscope, the structure of bacterial cell is look like a capsule. It has following components like outside the cell membrane and internal to cell membrane. This picture shows the ultrastructure of bacteria. Next is the capsule. It is the outer layer of the bacteria cell. Depending on the chemical nature, capsules are thick or thin, and rigid or flexible. It is most important virulence factor of bacteria which is visualized by negative staining technique. Generally, it composed of 98% water and 2% polysaccharide or glycoprotein, polypeptide or both. There are two types of capsules. 1. Macrocapsule. Thickness of 0.2 jm or more, visible under light microscope. 2. Microcapsule. Thickness is less than 0.2 jm, visible under electron microscope. Functions. 1. Preventive nature. Capsular polysaccharide binds significant amount of water, making cell resistant to drying and also prevent attachment of bacteriophage on cell surface. 2. Protection. It protect from mechanical injury, temperature, drying, etc. 3. Attachment. Capsule helps in attachment on the surface. 4. Example: Streptococcus mutans that cause dental caries attach on teeth surface by its capsule. 4. Antiphagocytic. Capsule resist phagocytosis by WBCs. 5. Source of nutrition. It acts as a resource organ of nutrition during insufficient nutrient supply. 6. Repulsion. Same charge capsulated. Bacteria repel each other. Cell wall. Cell wall is the rigid wall situated outside of the plasma membrane. It provides shape of the bacteria as well as provides protection from osmotic lysis. Cell wall is made up of peptidoglycan. The gram-positive bacteria contains thick homogeneous peptidoglycan or moraine layer, 20 to 80 nanometers thickness, outside the plasma membrane, whereas gram-negative bacteria has 2 to 7 nanometers. Peptidoglycan layer surrounded by 7 to 8 nanometers thick outer membrane. Peptidoglycan is a polymer, consists of two sugar molecules, viz. N-acetylglucosamine and N-acetylmuramic acid. Apart from that, it also contains several amino acids. Flagella. They are long, whip-like appendages found on the surface of many bacterial cells. They play a crucial role in bacterial motility, allowing bacteria to navigate their environment effectively. Role of flagella in bacteria. Flagella enable bacteria to move towards favorable conditions, such as nutrients, and away from harmful environments through a process called chemotaxis. They also aid in surface attachment, biofilm formation, and can play a role in pathogenesis by facilitating the invasion of host tissues. Fimbriae and pili. They are hair-like appendages found on the surface of many bacteria. They play crucial roles in bacterial adhesion, allowing bacteria to attach to surfaces, host tissues, and each other, which is essential for colonization and biofilm formation. Pili, often longer than Fimbriae, are also involved in processes such as DNA transfer during bacterial conjugation. Both structures are vital for bacterial pathogenicity and survival in various environments. Cell membrane. Cell or plasma membrane is a thin structure that completely surrounds the cell. It is about 8 nanometers thick. This structure is critical barrier separating the inside of cell from environment. The cell membrane is also highly selective barrier enabling the cell to concentrate a specific metabolite and excrete waste material. Mostly biological membrane is composed primarily of phospholipids, about 20 to 30%, and proteins, about 60 to 70%. The main function of cell membrane is selective permeability and transport of solute, electron transport and oxidative phosphorylation in aerobic species, excretion of hydrolytic exoenzymes. Inclusion bodies. Various types of inclusion bodies, organic and inorganic granules, are present in the cytoplasmic matrix. They are mainly used for storage of energy and reduce osmotic pressure by tying up molecules in particulate forms like polyphosphate granules, glycogen granule, etc. Ribosomes. Ribosomes are loosely attached to the plasma membrane, 70S ribosomes. They are made up of both protein and ribonucleic acid. They are the site of protein synthesis. Nucleoid. Prokaryotic chromosome is located in an irregularly shaped region called nucleoid. Nucleoid is composed of 60% DNA, 30% RNA and 10% protein by weight. Prokaryotes contain single circle of double-stranded DNA, but some have linear DNA chromosome. Mesosome. Mesosomes are present in the cell membrane of bacterial cells that fold inward. They play a major role in cellular respiration. They appear as a pocket formed by the cytoplasmic membrane, filled with vesicles, tubules or lamellae. This image is showing the morphological classification of bacteria. As per the morphological basis, bacteria are classified into 6 major groups which are depicted as follows. Next is true bacteria. They are a major group of classification which is further divided into 2 subgroups, namely Cocci and Bacilli. Cocci are round shape and Bacilli are rod-like structure. Actinomycetes. They are rigid organism but look like fungi. They exhibit branching and tend to form filaments. Basically they are gram-positive, but several species have complex cell wall structures that make the gram staining unsuitable. This slide is showing the spirochetes. They are double membrane bacteria, most of which have long, non-branched, helically coiled cells. They are gram-negative and are chemoheterotrophic in nature. Mycoplasmas. They are the smallest bacterial cell and not having rigid cell wall. They are indefinite shape and highly pleomorphic, alter their shape or size in response to environmental conditions in nature. Example: Mycoplasma pneumoniae, which causes pneumonia. Rickettsia. They are non-motile, gram-negative, non-spore-forming, small intracellular and pleomorphic bacteria. They are also present as Kochi, 0.1 micrometers in diameter, rods, 1 to 4 micrometers long, or thread-like, 10 micrometers long, forms. Rickettsia species are the pathogens responsible for typhus, rickettsial pox, Boutonner's fever and are susceptible to antibiotics of the tetracycline group. Examples: Rickettsia conneri, Rickettsia typhi, etc. Chlamydiae. They are ovoid in shape and gram-negative bacteria. Their cell wall is consisting of peptidoglycan and other proteins. They are very small, obligate intracellular parasites. They multiply in the cytoplasm of their host cell by a distinctive developmental cycle. They commonly infect humans or eye disease. Examples: Chlamydia trachomatis, causes trachoma eye disease. I request to you, please subscribe this channel and like and share this video to get more videos and updates. Thank you.